Hello everyone, good afternoon and welcome to today's class. So we're gonna start in 5 minutes so that people can join in. Okay. Okay, I guess we can start now. So we're not gonna start any new topic, as you know that I prefer to have regular revisions of whatever we have covered, so we can understand that where we are going, and we should not feel lost in the topic, because chances are there might be too much new information coming your way and you might feel that you are lost, right? So in this next session, in the first session we talked about the membrane and the proteins and the lipids. Now with respect to the membrane proteins, we are trying to understand proteins itself, right? Because they are the ones which provide the functionality to a membrane. So we are trying to understand whatever we know about the function. So to know the function, there has been a study about how the structure relates to the function of a protein. So from there we are trying to understand the structure of a protein. So from primary structure, secondary structure, tertiary structure, which is the functional form of the protein, we are at the secondary structural level right now. Okay, so if you remember that this was the paper by Pauling, who came up with the alpha helical secondary structure, which is one of the predominant secondary structure present in the proteins. In 1951, the title of the paper was The Structure of Proteins, Two Hydrogen Bonded Helical Configurations of the Polypeptide Chain. So the overall summary of the paper which they proposed was two hydrogen bonded helical structures of a polypeptide chain have been found in which the residues are stereochemically equivalent. The intra-atomic distances and bond angles have values found in amino acids, peptides, and other simple substances related to proteins. And the conjugated amide system is planar. In one structure, with 3.7 residues per turn, each carbonyl and imino group is attached by a hydrogen bond to the complementary group in the third amide group removed from it in the polypeptide chain. And in the other structure, with 5.1 residue per turn, each bonded of the fifth amide group. So now we know what do we mean by third amine group and fourth residue. It means that in alpha helix the system is N plus 4, N plus 3, sorry, and in other type of helix the system is N plus 5. So we'll look a brief introduction to secondary structures. Give me 2 minutes with respect to that. Let me share my screen. Yeah, I hope you guys can see it. Let me skip the ad.
We're going to talk about the structure of proteins. And what basically is a protein? A protein is a polymer consistent of many amino acids. So each amino acid represented by the circle is a monomer that forms the protein. Whenever you have many amino acids, it's also called a polypeptide. And the bond that connects each individual amino acid residue is a peptide bond. So let's talk about the individual structure of an amino acid. So an amino acid has a chiral carbon with a hydrogen attached to it, and it has an amine group attached to it as well. And then it has an R group and a carboxyl group. Now I'm going to draw another amino acid right next to it. The left side, where the nitrogen is located, is the N-terminal of the amino acid. The right side, that has the carboxyl group, is the C-terminal of the amino acid. Now, these two amino acids can react with each other in a condensation reaction where we're going to lose water, and we're going to connect the two amino acids into one molecule. So once water is removed, we could form a peptide bond. This is also called a dehydration reaction because you're losing water. And so this is going to be the new product of the reaction. So let's see if I can fit it in here. So this oxygen is gone. So now we have a nitrogen. Let me take out H2O. We have one hydrogen left over. We lost two once we lost water. So this is a dipeptide because we have two amino acids combined. And so this amide bond that we see here, that is the peptide bond. We formed a bond between the carbon and the nitrogen atom. So whenever you combine two amino acids to form a peptide bond, you're forming a covalent bond, which is hard to break. Now let's talk about the different levels of protein structure. There's 4 levels that you need to know: the primary structure, the secondary structure, the tertiary structure, and finally the quaternary structure. Now the primary structure is simply based on the sequence of the amino acids found in the protein. The sequence determines the shape of the protein. If you replace just one amino acid with another, it will completely change the shape of the protein. So the shape and its function is primarily determined by the sequence of amino acids. The secondary structure describes the localized shape of a protein. And there's 2 of them that you need to be familiar with: the alpha helix and the beta pleated sheet. In the alpha helix, you could see that it's stabilized by hydrogen bonds. The NH group of one amino acid interacts with the carbonyl group of another amino acid. And each turn contains about 3.6 amino acid residues. Now here is a visual illustration of the beta pleated sheet. Just like the alpha helix, it too is stabilized by hydrogen bonds between the carbonyl group of one amino acid and the NH group of another amino acid. Next up, we have the tertiary structure, which represents the 3-dimensional complete folding pattern of the protein. And so here's a visual illustration of it. So we can see some areas it's just a straight chain. Here we have an alpha helix, and here we have a beta pleated sheet. Now, the tertiary structure is one individual subunit. When you combine multiple subunits, you create a quaternary structure. So hemoglobin is an example of that. It has 4 individual subunits, 2 alpha subunits and 2 beta subunits. So hemoglobin is a protein with a quaternary structure. And so that's basically it for this video. Hopefully it gave you a basic understanding of proteins and the 4.
Right, so this was a very basic introductory video, which even at that time when we were discussing this topic for the first time, I wanted you guys to look at it, because it gives a very brief idea about what proteins are and from where the actual structure component comes into play. Right, one can also ask that why we are so intrigued with the structure of a protein. Why can't we be more fascinated only about the sequences? Because getting a sequence of a protein in comparison to structure of a protein is comparatively easy. But the main reason that we are so fascinated by the structure of protein is that the structure is oftenly correlated with the function of a protein. So if you want to understand the functionality of any protein, we should have an in-depth idea about the protein structure. So imagine you have a alien organism, right? You don't know anything about it, but its genome is also like us. Like we can see there are ATGC pairs, there are proteins and all that. But in reality, you won't know which protein is doing what in that new alien organism, right? So on the first hand, what will you do? You will try to see whether some of its protein have some similarity with respect to our protein, right? So imagine we have 20 residues, right? 20 amino acid residues. That organism, we don't know what those residues are and how is it, but at the end it's a protein, and that protein, it has some structure associated with it, but again at the end of the day we don't know what the sequence of the residues are there in the new organism. So can we somehow relate, for example, if a protein is looking like a barrel shaped protein, so we also have barrel shaped protein, so can we say whatever the function our proteins which are barrel shape are performing, that alien protein might perform similar function, or maybe in the same spectrum of the functions it might be performing, right? So that is why understanding the structure actually give us a more in-depth knowledge about the functionality of that protein, and obviously if you know the function, at the end we can alter its functionality or things based upon the understanding for therapeutic purposes, right? That is why understanding the 3D structure or the functional structure of protein is very important. So the another structure of protein which is predominantly present in at physiological condition is the beta sheet, right? So the discovery of the alpha helix and beta sheet, the principal structural features of proteins. So this was published in, I guess, PNAS in I guess very late 2003. It was like a commentary on Pauling's paper which we saw in the first slide. Okay, so what they mentioned is David S. Lindbergh, what he said that PNS papers by Linus Pauling, Robert Corey and Herman Branson in the spring of 1951 proposed the alpha helix and the beta sheet, now known to form the backbones of tens of thousands of proteins. They deduced these fundamental building blocks from properties of small molecules, known both from crystal structure and from Pauling's resonance theory of chemical bonding that predicted planar peptide groups. Earlier attempts by others to build models for protein helices had failed, both by including non-planar peptide and by insight on helices with an integral number of units per turn. In major respects, the Pauling-Corey-Branson model were astoundingly correct, including bond lengths that were not surpassed in accuracy for more than 40 years now. However, they did not consider the hand of the helix or the possibility of bent sheets. They also proposed structures and functions that have not been found, including the gamma helix. And so they are Pauling, Corey and Branson. So Pauling and Corey actually commented that we have been attacking the problem of the structure of protein in several ways. One of these ways is the complete and accurate determination of the crystal structure of amino acids, peptides and other simple substances related to proteins, in order that information about interatomic distances, bond angles and other configurational parameters might be obtained that would permit the reliable prediction of reasonable configurations of the polypeptide chain. Okay, this is the X-ray diffraction of DNA and this is the X-ray diffraction of the first protein, that is beta keratin. So basically, in short, if we know the structure of a protein, what we can say is that stereochemistry is important in determining which helices are possible, and integral symmetry has no role whatsoever. Now if we talk about one of the predominant secondary structure which is present in biological system at physiological condition is the alpha helix. So when Pauling, Corey and Branson constructed helices with planar amide groups with the precise bond dimensions they had observed in crystal structures and with linear hydrogen bonds of length 2.72 angstrom, they found there were only two possibilities. These two they called the helix with 3.7 residues per turn and the helix with 5.1 residue per turn, soon to be called the alpha helix and the gamma helix. The idea of the non-integral alpha helix had come to Pauling 3 years before when he was visiting professor at Oxford University. He got cold in the damp weather and spent several days in bed. He recalled that he was soon bored with detective novels and I didn't have any molecular model with me in Oxford, but I took a sheet of paper and sketched the atoms with the bonds between them and then folded the paper to bend one bond at the right angle. What I thought it should be relative to the other, and kept doing this, making a helix until I could form hydrogen bonds between one turn of the helix and the next turn of the helix, and it only took a few hours of doing that to discover the alpha helix. Correct? So this is how the alpha helix structure looks like. So as you know, it's a helix with pitch of 3 point, like in each turn there are 3.6 residues, and in alpha helix the first residue is hydrogen bonded to the fifth residue, that is there is n+4 pattern of bonding, and this is the Ramachandran plot which we have discussed quite in detail during our earlier sessions, so I'm not going to go again deep into it. We might see a small video lecture describing it again if you guys want it, as this is just a revision. So alpha helix, so the properties of alpha helix is 3.6 residues per turn, translation per residue is 1.5 angstrom, translations 5.4 angstrom per turn. Okay, so there is a hydrogen bonding between the carboxylic acid CO with the amines NH, which is I plus fourth residue. The phi and psi angles are the dihedral angles. So the phi angle is at minus 57 degrees and psi angle is at minus 47 degrees. The preference of the residues in helix is there. So there are certain amino acid residues which prefer to be in alpha helix, while there are certain residues which do not prefer to be in an alpha helix. Okay, on an average a helix length is around 10 residues. The helix have amphipathic nature, that is they have one side hydrophilic. They have both the properties of hydrophilic and hydrophobic. How we come to know this? We came to know when we actually saw the helix, the distribution of residues in the helical wheel representation. So when we see the helical wheel representation, so you can see the center point is the axis which is towards you, coming away out, like coming towards you from the screen. You can see one side is purely hydrophobic while the other side is purely hydrophilic, which means this helix has the property of both being hydrophobic as well as hydrophilic, right? And as the residues are placed like this, there is a dipole moment associated with the alpha helix. So dipole moment is basically the culmination of polar charge or the charges, and it helps in basically providing the non-covalent interactions when it is present in other macromolecules. Okay, so this is some basic information with respect to the alpha helix. We have discussed more about it, but I guess let me share this part of the slides so that we can recall whatever we have studied. Just give me 2 minutes. Yeah, I hope you guys can see my screen. Let me skip the ads. In the meantime, I will really appreciate if you guys can go on Moodle platform and check out the videos so that you can recall the topics and mail to me if you have any queries of any type. Okay.
We'll talk about alpha helix structure, which is a common secondary structure of protein. If we talk about protein structure, then it can be classified into 4 different levels: the primary structure, secondary structure, tertiary, and the quaternary structure. But this video would focus on the secondary structures. Out of the secondary structures, we have alpha helices, beta turns, and beta pleated sheet. But today's focus would be totally on alpha helix, its structure, its function, where we find them, and how we can study the structure of alpha helix and the factors that stabilize it. So this video is all about that. So if you haven't yet subscribed to my channel, hit that subscribe button and stay tuned till the end of this video. First of all, let's try to understand where do we find alpha helix structures. It can be found in many proteins, such as proteins that are receptors, and very common example is G protein coupled receptor. It has 7 transmembrane domain and each transmembrane domain is alpha helix, spanning 10 to 12 amino acid residues. Many transcription factors such as leucine, which contains leucine zipper motif, that is also an example of alpha helix. So many transcription factor, receptor, ion channels, and many other molecules might have alpha helix conformation. Now let us try to understand how we can study and gain the information about alpha helix. First, we have to start with X-ray crystallography, which begins with crystallization of the protein. Then there is X-ray crystallographic method where we obtain a diffraction pattern while we pass X-ray sample through this crystal. From this diffraction pattern, we can convert it into frequency domain to get the electron density map. After Fourier transformation, we get this electron density map. Once we get the electron density map, it's like the overall outline of a puzzle. And we have to fit our molecule of interest and start building model of it. And ultimately that model might give rise to the protein structure or structure of any molecule. So this is the overall workflow of X-ray crystallography by which we can understand the protein structure. If you want to know more about X-ray crystallography, click the link in the I button. So the biggest feature of alpha helix is that helix, and it was found by Pauling and colleagues around 1940s. So alpha helical structure has amino acid in specific orientation. And obviously you can understand the orientation is a helix fashion, where almost 4 amino acids are there per turn. 3.6 to be precise. So alpha helix has 3.6 residues per turn of the helix. All of these residues that are present in the alpha helix are bonded or interacting with each other via hydrogen bonding. And this hydrogen bonding does not take place in a haphazard manner, and there are certain rules that are governing which residues would form hydrogen bonding. Before coming to that rule, we should understand that from a scattered structure of a protein, that means the primary structure, making a geometrical structure like helix is a long process. It requires a lot of planning and organization to make such of geometrically organized structure. So there must be some rule which is governing these interaction, which helps to stabilize these kind of helical structures. Let us understand this rule. There is a rule in hydrogen bonding which is ensuring this helical structure is stabilized. Let's imagine a particular amino acid from the N-terbitus and we name it I. Then the next one would be I plus 1. After that, there would be I plus 2, I plus 3, and I plus 4. It turns out the hydrogen bond would be formed between I and I plus 4 amino acid. So the carboxyl group of I and the NH group of the I plus 4 amino acid would form hydrogen bond with each other. Irrespective of the position, this kind of rule is valid throughout the helix. Now let us talk about few special cases when helix is forming. Specific residues such as glutamate are good for helix formation. But if glutamate residues are there in repetition, then it might be bad because glutamate residues at pH 7 has negative charges, and these negative charges could cause strong electrostatic repulsion and thereby destabilizing the helix. The same goes for the basic amino acids, which are lysine or arginine. They are positively charged in pH 7. That is why they can also undergo electrostatic repulsion if they are very close to each other. But if they are dispersed throughout the structure, it might not be a problem. It would be actually favorable in terms of helix formation. Other amino acids such as proline is also termed as helix breaker, because once proline is present in a secondary structure, it would create a tilt in the secondary structure, as you can see here. And the amide hydrogen of proline cannot be used to make hydrogen bonds. And proline side chain interferes sterically with the backbone, and that is why it leads to a tilt of 30° corresponding to the helix axis. So obviously we can understand, if we have a proline residue in the secondary structure, it would be ultra destable. It would be destabilizing the helix. So that is why proline is known as helix breaker. Residues in an alpha helix typically adopt specific dihedral angles, and these dihedral angles are really important to define a particular structural characteristics. And the map of dihedral angle is basically the Ramachandran plot. In this Ramachandran plot, these helix falls around -60 to -45 phi and psi angles. So helix would be corresponding to this particular region, and there are different regions for other particular secondary structures such as beta pleated sheet or right-handed or left-handed helix. Coming to the helix sense, helix could be right-handed or left-handed. Right-handed helix are the most common ones found in biological system. It's very easy to understand right-handed helix. Try to point your thumb towards the helix axis, and your curled finger's direction would tell you the sense of the helix. Now, alpha helix, which is left, which has a left helicity or a right helicity, would be found in different locations in a Ramachandran plot as depicted here. Also, there are helix propensities. That means the probability of an amino acid to be found in a particular helix. This governs many factors. That means which of these amino acids might be incorporated to form a helix structure. Amino acids like alanine, leucine, methionine are helix makers. That means if these amino acids are present in a protein sequence, they would readily form alpha helical structures. Whereas other amino acids such as glycine and proline are known as helix breakers. They break the helix, but they are very good in terms of beta turn. That is why helix propensity index tells us by a particular score that what is the probability that one particular amino acid would form a helix. Now let me tell you, these alpha helix structures have a lot of peptide bonds, and each peptide bond might have slightly dipole interaction among them. So alpha helix acquires a net positive charge in one side and negative charge in other side due to dipole interaction, and these charges need to be neutralized in order to gain stability, and this is done by specifically negatively charged residues in the N terminus and positively charged residues in the C terminus. Positively charged residues such as arginine and lysine would help to neutralize the net negative charge, whereas negatively charged amino acid would help to neutralize the partial positive charge due to dipole, and this ensures the helix becomes stable. So let's just summarize the factors which are affecting helix stability. First of all, electrostatic repulsion. Second, bulkiness of adjacent R groups of amino acids. Hydrogen bonding between I and I+4 amino acid is absolutely crucial. Presence of proline or glycine really destabilize the helix. So these are helix breakers. And lastly, interaction between amino acids and the residues at the end determines the helical stability. So the alpha helical structure can be studied nicely with
Yeah, so this was the overview of alpha helix, and then we actually switched in the lectures when we were taking it for the first time. We actually switched towards X-ray crystallography and all that. So we not gonna go completely deep into it because we have already discussed most of it. Then about the left-handed alpha helix and proteins. So generally, majority of the time in the biological system, the right-handed alpha helices are present instead of left-handed alpha helices. Okay, so some basic. Let me get to the structure. Yeah. Some basic pointers about the structure of a protein. So it goes like, proteins are big molecules. The covalent backbone of a typical protein contains hundreds of individual bonds. Because free rotation is possible around many of these bonds, the protein can, in principle, assume a virtually uncountable number of spatial arrangements or conformations. In reality, however, each protein has a specific chemical or structural function, which suggests that each has a unique three-dimensional structure. Protein structures are stabilized by non-covalent interactions and forces. Formation of a thermodynamically favorable structure depends on the influence of the hydrophobic effect, hydrogen bonds, ionic interactions, and van der Waals forces. Natural protein structures are constrained by peptide bonds whose configurations can be described by the dihedral angles. Protein segments can adopt regular secondary structures such as the helix and the conformation. These structures are defined by particular values of phi, and their formation is impacted by the amino acid composition of the segment. All of the phi and values of a given protein structure can be visualized using Ramachandran plot. Tertiary structure describes the well-defined three-dimensional fold adopted by a protein. Protein structures are often built by combinatorial use of common protein folds or motifs. Quaternary structure describes the interaction between components of a multi-subunit assembly. Tertiary structure is determined by amino acid sequence. Even though protein folding is complex, some denatured proteins can spontaneously refold into their active conformation based only on the chemical properties of the constituent amino acids. Cellular proteostasis involves numerous pathways that regulate the folding, unfolding, and degradation of proteins. Many human diseases arise from protein misfolding and defects in proteostasis. The three-dimensional structure of proteins can be defined. Structural biologists use a variety of instruments and computational methods to solve biomolecular structures. The choice of method may depend on factors such as the size of the protein being studied, its properties, or the desired resolution of the final structure. The possible conformations of a protein or protein segment include any structural state it can achieve without breaking covalent bonds. A change in conformation could occur, for example, by rotation about single bonds. However, of the many conformations that are theoretically possible in a protein containing hundreds of single bonds, one or a few generally predominate under biological conditions. The need for multiple stable conformations reflects the changes that must take place in most proteins as they bind to other molecules or catalyze reactions. The conformations existing under a given set of conditions are usually the ones that are thermodynamically the most stable, that is, having the lowest free energy. Proteins in any of their functional folded conformations are called native proteins. Stability is the tendency of a protein to maintain a native conformation. Native proteins are only marginally stable. The delta G separating the folded and unfolded states in typical proteins under physiological conditions is in the range of only 5 to 65 kJ per mole. A given polypeptide chain can theoretically assume countless conformations, and as a result, the unfolded state of a protein is characterized by a high degree of conformational entropy. This entropy, along with the hydrogen bonding interactions of many groups in the polypeptide chain with the solvent, tends to maintain the unfolded state. The chemical interactions that counteract these effects and stabilize the native conformation include disulfide bonds and the weak non-covalent interaction and forces. So the remaining revision we will do in the next class. It's very few slides are left, and then we can continue our discussion. I'll show you one of the simulations of a protein which is transporting a substrate, so you can get an idea about how exactly the proteins look like and how it is functional. Okay, so thank you so much for coming in, and hope to see you in the next class. Thank you.